doke. So, asbestos. What do you know about asbestos?
So asbestos is a group of naturally occurring minerals, and they group together, and they're resistant, heat resistant, and they don't conduct electricity, and it leads to basically a new type of lung cancer.
Okay, so for our procedure, what we had was we had 5 boxes, and these boxes all contained 100 mice. Within this, we had different levels of asbestos in the air. The first one is our control one, and it has 0 grams in here. So we'll see how much lung cancer just is by chance. And then the second box contains how much? 15 micrograms per liter of air, per square liter of air. And then we continually went up by significant amounts by going from 15 to 150 to 500 to 1,000 micrograms per liter. And by exposing all 100 mice to the same amount of asbestos per liter of air, then we can find the average or percentages of the lung cancer that will develop due to the asbestos. And for this experiment, we made sure that all the boxes were at 60 degrees, and we left them in there for 3 months. And then we took them out and put them in a regular, normal sort of habitat setting. And then we waited 9 months, and after 9 months, we killed them. And when we killed them, we then cut them open and look at their lungs to see if they have any tumors or lung cancer.
So these are our results. So as you can see, the higher the concentration of asbestos, the more likely the mouse is to, or the lung is to get cancer. And the results were kind of surprising because we expected a lot more to have it than actually it turned out to be. But you can still see the gradual increase as the asbestos amount grows.
And what does this mean? It means that inhaling asbestos may lead to causing cancer, and the more exposed you are to it, the greater your chances. But I think one of the things that maybe might have made our results less is not really knowing how much of a microgram in a liter is. I'm not really sure, what the average exposure to asbestos would be, so that might be good for next time. And the next step is exposing asbestos to human lung cells, just so we can see the difference between them and mice. And the asbestos will be put into a solution which will be absorbed by the cells. And the different cells will put a different amount of asbestos in each solution, so we still get the variety we had with the mice. And we will observe the results of the cells. Any questions?
Okay, so why did you guys decide to go from an animal study to a cell study? What was the factor? What was the sort of question that you're trying to look at by looking at a cell study next?
How it would be different from humans.
Okay, so you think you had some good results from animals, and so now you're trying to look at human cells.
So it's kind of like a way of double checking that it wasn't just mice lungs that would absorb the asbestos, because if in fact that the mice, their lungs were different than ours and that they could absorb asbestos and the human lungs couldn't absorb asbestos, that would mean that the chance of cancer, if we couldn't, would be 0 due to exposure to asbestos. So by double checking on a human lung, we'll make sure that it is plausible for a human lung to absorb asbestos.
Any other questions about how they decided to do their experiment or what they pulled from it? Okay, nice job, you guys.